Well, welcome everybody to the Aloha Friday edition of the Hawaii Book and Music Festival panel here, and we have some great guests for you. So today our panelists are going to be focusing in on a couple of key topics that are top of mind for a lot of us in the community. One is a COVID-19 vaccine, and the other one is a COVID-19 test. So with that, I will give each of our panelists a chance to share some of the work that they're doing. And we'll start off with Axel, and he is involved with the actual COVID-19 vaccine process.
So I'll just tell you a little bit of the story, and this is really kind of, where it started. The question was earlier this year, is a vaccine actually possible to protect against COVID-19? Because the virus is actually out of the family of coronaviruses, where there are no existing vaccines at this moment there's no vaccines available on the market for any coronavirus. Then some of our colleagues in the mainland were testing if actually a infection with the coronavirus will cause immunity that then can protect against the challenge. And this is what's published in May. So, yes, a vaccine seems possible, but it is not necessarily going so fast. So one of the things that we have to look at when we develop a new vaccine is we have to select a proper antigen. The antigen is a protein that the virus or other pathogen makes that then will actually generate an immune response that will protect against the pathogen. And what's important here to remember is immunity or an immune response is not necessarily, or it does not equate with protection. So very important for diagnostics is also the question, is it actually going? So in this case, now we're looking at an antigen that will actually protect you from a subsequent infection. And the spike protein is the protein for coronaviruses that will do this. The spike protein mediates the virus receptor binding and also membrane fusion. Therefore, it allows the virus to infect a cell. And so that's what all coronavirus vaccines are now focusing on, is to protect this, to prevent this future infection. The spike protein is actually a very large protein. It's a class 1 fusion protein that exists as a trimeric protein on the surface of the virus. And it is a metastable pre-fusion state. That means that after it fuses with the membrane, the protein changes its shape dramatically. This is the pre-fusion formation that you see on the surface of the virus. And actually it makes these very nice electron micrographs that we frequently see in the media these days. The protein consists of 2 different subunits, and we have learned how we can lock them together and how we can also keep them in this pre-fusion stage, which is very important because the post-fusion one is not going to give us this full protection. So here's just a quick overview of all the different versions of vaccines that are possible and that are one way or another under development. What we see up here on the top is a live attenuated vaccine. Live attenuated vaccines, very similar to the actual coronavirus, will infect a cell. The virus will then replicate in there. It will release more viruses, and they will be presented to the immune system. An inactivated antigen will work the same way. And then down here, we have the friends of the RNA and DNA vaccines. So that is where Moderna and Pfizer sit. Those basically program the cell to make this spike protein that we just talked about. Other ways to deliver the genes would be viral vectors. There's replicating ones. Merck is working on a vaccine like that, and non-replicating ones. This is where we find the likes of Johnson & Johnson and AstraZeneca. Virus-like particles and subunits are other approaches. Subunit vaccines in advanced stage development are with Sanofi and Novavax, and then also here at JABSOM, but we're not quite that far yet. So now all of these antigens are going to be presented to this kind of green cell here. This is what is called the antigen presenting cell, and that will then talk with the immune system, with the T cells and the B cells, and form a nice immune response, hopefully. There's a total of over 200 vaccine candidates that are under development right now, and I'll just leave it at that. We're one of them that is being tracked by the Milken Institute COVID-19 tracker. It's a nice website if anyone is interested. Our platform is based on a highly productive insect cell expression system that we have used for decades now to generate high quality antigens. One of the specific characteristics is that we use stably transformed cell lines. And then what we have shown is that we can produce uniformly glycosylated proteins. Cells are sometimes a little bit moody, and they may, depending on how they feel, they may, glycosylate proteins more or less. That means they put sugars on the surface, and those sugars are very, very important so that the protein forms the folds in the proper way. And then what we combine this with this immunoaffinity chromatography to very specifically purify these proteins. So we have a different purification method for every target. This is how the process looks kind of schematically. We have an antigen production bioreactor. We also produce our antibody that we typically use for the purification, and then we use our FPLC purification system. And then what we've shown is that this purification process seems to be key to maintain proper immunogenicity and efficacy. And in that, we're differentiating from other people using a similar platform. This is just an example. This is a single-step purification process where we can show from a supernatant that has a lot of different proteins, the different stripes here, going to a very pure product here in a single-step process. We're then combining this with a thermostabilization process, which we work on with the mainland biotech company. And what we have shown is that we can actually lyophilize, which is then basically kind of your long-term stability processing. We can lyophilize the protein together with the adjuvant that is needed to get the proper immune response. We just recently published a paper on this for our filovirus glycoprotein vaccines. Those are in development for Ebola and other filoviruses. Now let's shift focus to the COVID-19 vaccine. We first tested whether our adjuvant actually produces the right type of immune response. So this is part of the platform. We need a protein, we need the adjuvant, and we need to be able to then thermostabilize this. So we combined, this was a model antigen, we combined this SARS spike protein with either the adjuvant we call Covaxin HT. There's also, we tested al-hydrogel, which is the standard adjuvant used in very many vaccines, and then also tested it without adjuvant. One of the things that was seen here is that with our selected adjuvant, we actually get a very potent response already after one dose. This is in mice. With 2 doses with alum or protein, we also have very good antibody titers. So this actually, I just want to mention, this work is led in my lab by Dr. Lai, who has been working with me for many years now on vaccine formulation, and really an excellent immunologist. And what we've shown as well is that there's cross-reactivity here. This is SARS coronavirus 2 is the causative agent of COVID-19. There's also SARS virus from the early 2000s that caused the outbreak. So there's actually going to be pretty nice cross-reactivity between these two. there is a possibility that the vaccine might protect against more than just against the current COVID-19, but it could be related viruses as well if, for example, the virus mutates. What's most important is virus neutralization. So with our Covaxin adjuvant, we have very potent titers. These compare to virus neutralizing titers in patients that have recovered from the COVID-19 disease. The other adjuvant hydrogel does not induce neutralizing titers. And then furthermore, also we have cell-mediated immunity. As you remember, the immune system has B and T cells, meaning now we have T cells that actually help our immune system recall, which is basically what we need to have a really effective vaccine. So we've shown potent humoral immunity, that is antibodies. We've shown that with the lead adjuvant, we generate high neutralizing antibody titers. We have shown that there's a strong Th1 type immunity. That's the one that we want, so that we don't get any kind of bad side reaction. And that this paper has just been accepted this week in Frontiers in Immunology, also by my team.
Now I'd like to invite Patrick Sullivan from Oceanit to talk about the work that they're doing with the COVID-19 tests, which is a very novel approach, and I think he's pretty far away along to perhaps even getting this implemented. Go ahead, Kat.
Thank you, Burt, and thanks, Axel. Very interesting. The thing with a vaccine or an antiviral treatment, you've got to use it in people eventually, and it takes a lot of time and it takes cash to do that. Whereas this is actually, the test has been quite a sprint. It still is expensive, but we have plans for antivirals, vaccines, and a variety of other things in the future. But what I'll do is I'll review how we think about it and what we do, and then where we are. But so as a backdrop, Oceanit is a mine-to-market company. It started about 30-some years ago. So we're in these buckets of energy, aerospace, life sciences, and information systems. And we've got a variety of ways to get things to market. So it's private equity, corporate co-development, and other modalities. We've invented ways to disintermediate venture capital because it's in short supply in Hawaii. But to get just in contrast, typical numbers, to get a therapeutic to market, 10 to 12 years, $100 to $150 million. To get a test, rule of thumb is 25 to 50 million, 7 to 8 years. And it's expensive. And of course, if you're doing a bunch of these things, it takes a lot of capital. So one of the issues as we talk about things here in Hawaii is without the sort of rethinking the whole capital issue, it's hard to look at moving products like this to market. But the thing we're doing is actually moving along. We're a mind-to-market company, so we've got the blue zone where we do deep science, curiosity, and discovery. We have about 200 people, maybe 100 PhD level scientists and engineers, and they operate a little bit like a university or a national lab. And then on the green side, the right side, the green zone, it's all about products. It's human-centered. How do we impact humans and society with products or other things? And we connect them to the rock and roll zone because they never quite match up. Usually on the science side, on the left, we think we know what we're going to do until you actually work with a customer, a human, you're out there in the market, you really don't know. And of the risk, it turns out there's technical risk, execution risk, market risk in any technology, but the biggest risk is market. It's about 50%. So getting the market right is a huge issue, and market timing is an enormous problem. We kind of do our work in this portfolio of interesting, challenging, and disruptive. So interesting are things anybody can do, challenging are things anybody can do, but are hard to execute. Disruption is where you're pushing the edges of what science allows. And that's our area. That's what we're really good at. And again, we've been doing this for decades, but we've gotten better and better at it. There's a process, which I won't go into, but I just show it as it's laid out here, called Intellectual Anarchy. And it starts with a really good question, and it kind of follows this graph over and over and over. It repeats itself. So it's another conversation, but that's the uncanny thing about it. But it starts with a question of what's interesting and important. So we move from ideas, programs, projects to products. Left axis is risk, right axis is benefit, x-axis is time. So the ideas can be very broad. So it's not limited by subject. It's really what's interesting and important. And then we have government support to reduce risk, and then we start talking to companies and can do venture or other things to get it to market. We collaborate with about 60 universities. University of Hawaii is a special university, and we have a long relationship with them. Lots of companies. And this has been an interesting thing. The more we do, the more we get, the more we do. It's kind of funny how it's working, but a lot of big companies. And what we discovered is we do things differently, and it addresses a gap in the marketplace, especially in the pipeline of technology. You can read about it in a book. It's in Barnes & Noble at All in One. It's on Amazon and other outlets, called Intellectual Anarchy. I started this a few years back, and it came out in August. And it's relevant specifically, too, because for what we're doing in Hawaii, it is a Hawaii story. So the journey to what we're doing with ASSURE-019, or the test, started with this conversation on Anthronoetic artificial intelligence. So human-style intelligence, small data, ambiguity, edge cases, not machine learning, which we've been doing for decades, not neural nets, which we've done for decades, but this style deals with things that humans are really good at and machines are terrible at right now. But it's actually, we started 6 years ago with, in our case, it was Office of Naval Research, which took a chance on this idea, and we've actually gotten some terrific results from that, and that work continues. It's just exploded into a whole bunch of programs. But this question of an application for biology started with this conversation around cancer and the question of causality versus correlation, which is framed here in this picture with, Plato and Aristotle, this debate, which continues today. So a lot of medicine is based on correlations, but what we're trying to investigate or consider is this idea of causality, the mathematical connection between what's going on in biology, particularly genomics and human disease, in this question of can we create these mathematical models? And we applied that to this strand, this idea of genomics. And if you look at nucleotides, we put together sequences, but we thought if an American alphabet is 26 letters, letters make words, words make sentences, and you have infinite complexity with sentences, perhaps you could apply the same thinking to understanding cells and the intention of cells. And if we could do justification, and we do justification in the AI space where we can sort of summarize, like I used the example of the play Macbeth by Shakespeare, with 17,000 words, machine learning will produce a word map we produce that can justify and say, well, it's about revenge, although revenge doesn't show up in the play. In this case, we produced the grammar of RNA as a tool set with DARPA. We started this about 3 years ago. That was very good and created a computational methodology. And with that, and then some collaboration with some folks at the University of Hawaii Medical School, we were able to go from the models to the lab, to the models to the lab, and kind of iterate to produce a molecule to address this. And anyway, so the sequence, this is part of the human genome, lots of these letters. The question was, can we consider it a language? This is the test we produced. So it's what's called a lateral flow assay, which is like a pregnancy test, except ours is based on spit. And you basically mix it with a reagent, and you get a line. And in our case, there's two lines. One says the test works. The other one says you've got it or you don't got it. Some of the early results That's a model of the molecule and the nucleocapsid proteins that it binds to. Some of the live virus testing here with this V1 to 3, which we did at University of Hawaii BSL-3 lab. So a lot of support there. But it's kind of an interesting idea because then you're synthesizing a molecule specific for a mission or a purpose. And so we talked about this in the middle of last year, this thing called digital medicine. So basically designing solutions for medicine based on mathematics as a way to create greater connection causally versus just, correlating 1 million people that smoke, 1 million people that don't, and see which ones die first kind of thing. So this gives you an idea. We're trying to make it a very cheap test. Numbers thrown around like $20. We think we can make it cheaper, get a result in 10 minutes. This curve on the right is an adaptation from this guy at Harvard, Mike Mina. And we put together early on some analysis on how the virus grows in this zone here. So I'm going to review this as we move further along, but we collaborated with a group of people on our analysis, but then adopted some of their approach to understanding where you are infectious. Not only Mike Mina, but this guy, Larry Moore and Roy Parker at University of Colorado Boulder. Our test looks something like this. We keep modifying it, but you collect sample of saliva. This is a little thing you stick in your mouth, and then you squirt it into a little cup. And one of the issues we've discovered is saliva is complicated. Not everybody can spit. Not everybody can spit on command. You can document the whole thing with a mobile phone app, which we've already got done. And we're going back and forth on different ways to collect saliva, which when we started it with our clinical partner at Queens, we discovered, not everybody can spit. But actually, the results have been really good there. They've been good to work with. And we're trying to now still simplify the whole thing of collecting spit. This shows you how it works. You collect it, you squeeze it in this thing, shake it, and you wait 10 minutes and you get a line, either 1 to 2 lines. And then we looked at the operationalization of it. So how do you use it to open the state? And this idea of bubbles and bubbles. And so we've been working on this in a conversation with the state, although I might add that there's some reluctance. I think the neighbor islands are trying to create bubbles. I think they have to. I think the state of Hawaii needs to create bubbles and grow those bubbles to make one big bubble. And I think that's how you control things here. This curve is an adaptation, again, of the work from Mike Mina, Larry Moore, Roy Parker, and they put the code online so you can run these simulations. It's very nice. You peak in about 5 days. So the left axis is infectiousness and from day of exposure. So you kind of max out 4 to 5 days, but you still may be producing virus. We figured that the way to do this then is when you walk off an airplane, you should spit in a cup, check it. If you're good to go, test every day for 5 days thereafter, kind of like a prescription. And you can use this in restaurants when, restaurant workers come in and check people when they come in the door to create a safe space. Again, you start with small bubbles and create bigger bubbles. Low cost, you track it with a mobile app. And it's all about this term of sample to answer time. 2 or 3 days is not good. There's some tests out there, rapid tests, where you can get maybe 45 minutes. They take time, you need equipment. But this is intended eventually to be an over-the-counter test. This is an expansion of that curve. It just kind of shows you pre-infectious when you're transmitting virus and post-infectious, where a PCR test will still produce a positive result, but you're not really at risk of infecting anybody. Just got some strand, RNA hanging around, which can persist. For some people, we've heard up to 60 days it shows up, but they're really not producing virus. They just got some, they get a positive result because the PCR test is very sensitive and looks for the sequence of the RNA. And this is an interesting rendition, again, based on these other guys' work that we've modified, but infectiousness is a function of days between testing. So if you test every day, you're going to drive it to close to 0. Test every couple days, you're in the 90s. So frequency of testing matters. The frequency of testing only works if it's really easy and inexpensive. And the last thing I thought I'd talk about is manufacturing. We intend to make some of this here. The idea of manufacturing gets a variety of interesting responses here. And I've explained to people, you know, these molecules are smaller than coffee beans, and they're smaller than sugar, you know, granules. They're the kinds of things you could produce here. But of course, you need infrastructure and other kinds of things. What we're looking at doing to make this broadly available. So we're looking at how do we produce 25,000 tests a day here in Hawaii, but we're looking at, for the national need, 1 to 5 million per day with different manufacturing partners, which we're talking to, and some international partners too. I think there's an enormous need for rapid testing.